All right, hi everyone. Welcome to the July Boulder Peptide virtual seminar. We're excited to have Dr. Jessica Kramer here to give a talk. And Kevin McGowan's gonna be the moderator today. Before we begin, I just want to let everybody know that today is the last day to take advantage of the early bird discounted registration rates for the live meeting in September. That meeting is going to be September 18th through 21st in Napa Valley, and we hope everybody will join us for that. So now I'll turn it over to Kevin.
Hello, everyone. Yeah, we are very excited to have Dr. Jessica Kramer here today to present to us. I'll get just a couple of logistics out of the way first. One, if you have questions, on the bottom bar, there's a Q&A tab. If you click that, you can ask your question. If you use the chat feature, that will pop up on the screen. So we're trying to avoid that, just to avoid the confusion. And then at the end, if you do have a live question you would like to ask, you're welcome to use the little hand raise icon, and Lauren can unmute you so that you can ask your question in real time. And so a background about Dr. Kramer. So she got her BS in chemistry from the University of Utah in 2004 from the lab of Professor Dale Poulter. And then she followed that with a PhD at the University of California in Los Angeles in Tim Deming's lab in the Department of Chemistry, after which she went on to her PhD or to the lab of Professor Carolyn Bertozzi at UC Berkeley and Stanford as an NIH postdoc fellow and a UC Chancellor's postdoctoral fellow. Her research efforts have been recognized with numerous awards, including the Henkel Award for Outstanding Graduate Research in Polymer Chemistry, Norma Stoddard Prize, Saul and Sylvia Weinstein Dissertation Award, Excellence in Graduation Polymer Research Award, and she even presented an experiment for the former Utah governor and U.S. presidential candidate, John Huntsman. So we are very excited to have you here, and I will turn it over to you.
Thanks so much, Kevin. Really excited to interact with the Boulder Peptide Group, which I actually haven't been to one of your meetings before. So I've got to get that on my calendar. I am really excited to talk about this particular project, which is a fairly recent work in my lab. And so I actually haven't given many presentations on this work before. So this is exciting. Today, the project I'm going to talk about is zwitterionic peptides that we use to stabilize protein therapeutics. And so before I get into that, I wanted to just kind of, I'm not sure how many folks are registered because I can't see your faces right now, but if you have students or postdocs, or you're looking for a place to come for research, I want to just give a brief shout out to University of Utah. It really is a wonderful place to do graduate school or even come for a postdoc. We have several graduate programs, both in my home department of biomedical engineering, but I also have students that come from a bioscience umbrella program that serves many chemistry and medical departments and pharmacy. And it's, of course, a very nice place to live with skiing and national parks. So keep us in mind. Always looking for great people for my lab. And one of our big projects is thinking about synthetic mucins and mucus. And I'm not talking about that today for once. That's a big focus of my lab. If that's an area of interest, keep us in mind. But the projects I'm going to talk about today are really thinking about new methods in biomaterials, synthesis, and applications. And as I mentioned, zwitterionic structures. And the origins of this project actually started from a collaboration with Danny Chow, who is now at Stanford, but used to be at the University of Utah, and is a good friend in the peptide and chemical biology world. And Danny was working with Matt Weber, who's at Notre Dame, and they had been studying insulin, in particular, how to formulate insulin so that it's bioavailable and stable. And so this was a new area of research that actually Danny brought to my attention, at first thinking about glycosylated structures like mucins, and we ended up thinking of something simpler and better, which is what I'll talk about today. And so this project is in collaboration with these folks. I know Danny interacts a lot with Boulder Peptide Society, and I think is an award winner in recent past. So lots of connections there. And the reason that Danny and Matt's labs are focusing a lot on insulin is that, for one, I think we all know this is one of the most important and impactful peptide drugs or drugs on the market. Incredibly important life-saving medicine. And one of the challenges with insulin is that it will form aggregates. And so it exists in this hexamer structure that is in equilibrium with dimer and monomeric structures. And this structure shown over here is actually the biologically active conformation. And so when the equilibrium lies the wrong way, of course, that is not productive for therapeutic use. And when the structure becomes partially unfolded, we can also end up generating oligomers and fibrils. And so these are aggregated structures that are not therapeutically useful and end up being sort of a waste of the drug. And this is really a big challenge for shipping and storage in particular. Just practical application of these drugs. I think if anybody knows a diabetic family or friend member, they're keeping their insulin in the fridge. And so cold chain storage is really essential, and not just for insulin, actually. This is a broader challenge as we continue to develop more and more peptide and protein therapeutics. So I'm talking a lot about insulin, but this is really just an example. I think we've all heard a lot about, for example, mRNA vaccines in the last couple of years, and the cold chain storage required for those types of therapeutics. So this is a challenge for insulin in particular, but also really there's a growing need to consider what happens during shipping and storage, particularly in developing countries where cold chain storage, refrigerated trucks are just simply not practical. So how can we address this challenge and get life-saving drugs into people's hands without degrading them? Okay, I wanted to just kind of give a shout out to like why we really need simple and cost-effective solutions and where the market is going with these types of therapeutics. And so this is not accounting for insulin, because that is obviously a way bigger market than other peptide drugs. This is a little bit old now, but from 2019, the sales in millions of US dollars and some of the candidates, and I'll actually refer back to glitterimer in a few minutes, which is an MS drug. The recombinant protein and antibody market are also really, really big and projected to grow tremendously in the coming decades. We have so many new antibody drugs for all kinds of human diseases, and there's really no end in sight for where that's going, which is amazing. However, all of these therapeutics are really used in cold chain storage at this point, if they are delivered in solution. And so we really need simple and cost-effective solutions to address broadly the issue of therapeutic stability, not just for smaller peptides, but also for recombinant proteins and antibodies. Okay, so what are people doing in this space? And kind of this is where I came in working with Danny and Matt in their work on insulin. And so PEG, which I would guess everybody listening today is probably familiar with PEG. The structure is shown here. It is a very hydrophilic, non-ionic repeat unit of ethylene oxide. It's kind of the oldest trick in the book for stealth coatings to stabilize, for example, nanoparticles or other items you don't want to be recognized by the immune system. But PEG has also been used as a stabilizer, and particularly for insulin, as an example. And this is an article header back from 23 years ago in the year 2000 from actually a U of U colleague, a former U of U colleague, Sung-Won Kim, who was very active in this area. And so they were some of the early adopters of thinking about attaching something to insulin to inhibit this aggregation pathway that ends up rendering it not therapeutically useful. So fast forward in the coming decades, Danny and Matt's work, they have used PEG in many different formulations to try to stabilize insulin. And so I'm going to show in just a second what this little ring represents and how they were using PEG. But at the time that we ended up talking with Danny and Matt, Danny actually, came to my lab and said, can we come up with something better than PEG? My lab has a big expertise in polypeptides, in particular glycopeptides. And so we thought, Danny thought first, maybe we could come up with an alternative to PEG that was glycosylated. And there's some examples of this, which I'll show in a few minutes. And the reason that people are interested in PEG alternatives is because there are actually many drawbacks. And I picked up this graphic actually from BioPharmaceuticals, which is a worldwide supplier of modified PEGs and PEG reagents. And so this sort of like limitations diagram actually comes from a PEG supplier, which is why I picked it. They did this press release back in 2019, put this on their website and said potential limitations of PEG-related therapeutic proteins. And so some of the limitations and why we need PEG alternatives is that PEG can, of course, if you attach something to a therapeutic or even a nanoparticle like drawn, you might inhibit intracellular uptake because now you've changed the whole properties of that particle or that peptide. You've changed the size and shape and molecular recognition and binding properties. So basic problem, you might inhibit the therapeutic value. And then from kind of the sort of downstream effects off target might be anti-PEG antibodies, and there have been numerous reports and publications showing that while PEG is relatively inert compared to things that are strong antigens, actually with repeated exposure and in some individuals, which like many things in the immune system, we still don't understand exactly why. There is a relatively, decent anti-PEG antibody response that over time becomes stronger and stronger and actually could be concerning. So there's that aspect. And then finally, PEG is just simply, it's not biodegradable. It actually can accumulate inside of macrophages, in vacuoles and other places where with repeated exposure, you can actually find inside of these macrophage cells just piles of PEG that sits there. So not very good. So we do need alternatives despite how useful PEG has been. Okay, so as we move into what we did as a PEG alternative, I wanted to stop and point out what that little ring was in the ways that Danny and Matt had previously used PEG to conjugate to insulin. And so here's this little ring, and this is representing CB7, which is this molecular ring structure shown over here on the left. And back in 2011, the Urbach lab showed something pretty remarkable for host guest chemistry with this ring structure. And so CB7 is really nice because it's water soluble, so useful for biological applications, but it has a nonpolar interior. So inside of this ring, there's actually just these kind of hydrocarbon or nitrogen lone pairs sticking in there. So we have this nonpolar interior, but the portal part here is called the portal, is lined with carbonyls and is actually quite polar. And so we have these two different properties, and based on the size and these electronic properties, the ring can participate in host-guest chemistry, specifically with aromatic groups that have a neighboring cation. And so they showed in this Urbach lab publication, a seminal work, they showed that this CB7 structure will very specifically bind to peptides that have an N-terminal aromatic group, such as phenylalanine or tryptophan or tyrosine. And the association constant is actually quite good. And comparison of, for example, an insulin structure, which has a terminal phenylalanine residue, comparing that to an insulin structure that is minus phenylalanine, the specificity is 1,000-fold greater. So this thing will essentially just stick onto the end of insulin with very high affinity. And what's really cool is that this complex is stable at concentrations relevant for packaging, shipping, storage. But upon use, an injection, for example, into a human, the complex dissociates. And so you are left with unmodified insulin. And so in this ring structure I showed here or that's used by the Chow and Weber lab, what they did is attach PEG to the CB7. And so they explored these stabilizing effects of PEG on insulin, but in a way that doesn't covalently modify insulin. And you can actually recover the unmodified therapeutic. So that's a really cool strategy, kind of pioneered by the Urbach lab and then developed for insulin stabilizing applications by my collaborators. Okay, so thinking about this strategy, but not using PEG, what have people done? Well, there's lots of strategies that people have explored. I'm going to highlight a few that have been particularly used for the insulin example, but there are many other PEG alternatives used for other strategies, maybe not related to insulin. So the kind of how the collaboration started between my lab and Danny and Matt is thinking about my lab's expertise in glycopolymers, because there had been some promising prior publications where things like glycosaminoglycans, which are repeating chains of sugar units, these are structures naturally found in the extracellular matrix. And so these structures had been actually covalently attached, so permanently modifying insulin, but showing stability to aggregation. And this was a really nice example with chondroitin and heparin. And then the Maynard lab at UCLA had shown some really nice data where they used methacrylate polymers with trehalose, this disaccharide here, trehalose, attached to insulin. And they had shown some very nice stabilizing effects. So 2 examples with glycopolymers, and these are kind of attractive, or they're nice because they're PEG-like in that they're not ionic, so no charge, but they're still highly hydrophilic. And depending on the structure of the sugar, I don't want to give a false impression that we can just say, oh, these glycopolymers are not immunogenic, because it really depends on what sugars are present. For example, in my undergrad class in physiology, I teach about blood types, and the presence or absence of one single sugar determines your blood type and whether or not you will have a life-ending coagulation event, right? So it really matters what sugar is there, but in general, there are many glycan structures that are very well tolerated by the immune system. So those are some attractive parts. However, there's some drawbacks to these previous approaches, which for one, they're usually relatively expensive to prepare. For example, these methacrylates that are modified with sugars, it takes some synthesis to do that. But also for the methacrylate example, it's again a non-native backbone that's potentially not biodegradable or might accumulate in some unwanted ways. So how can we maybe do better? Well, so in talking with Danny and Matt and kind of thinking about my background and expertise in polypeptide synthesis and properties, I proposed something different, which was zwitterionic polymers. And so like PEG and like glycopolymers, the overall net charge is 0. They are neutral. However, that is, you got to look at the fine print because zwitterions have both a positive and a negative charge. And so they're overall neutral, but these opposing charge properties typically give zwitterionic polymers very good water solubility. So that's kind of the part there, not ionic overall, neutral, but good water solubility, so similar to PEG and glycopolymers. And in some head-to-head comparisons of PEG, zwitterionic polymers had actually performed very, very well in a broad range of applications. There is a lot of interest in nanoparticle or drug delivery coatings replacing PEG with zwitterions. And so there's actually a lot of data from those types of studies about protein absorption, like from serum, and nanoparticle aggregation. And so in head-to-head comparisons, zwitterions actually perform really well compared to PEG. Also in long circulation times in vivo work and lower immunogenicity than PEG, and I put it in parentheses so far, right? Because we actually have many decades of data on PEG, but less so on zwitterionic polymers. So I think we need to be open and curious as to what the response will be to some of these structures. However, in terms of the application of these types of structures, there are a few examples to insulin. And again, they're typically based on non-natural repeat units. I've shown this review here where there are a couple of different zwitterionic structures that were appended usually covalently to insulin, so permanent modification of the therapeutic. And then again with a polymer that is maybe not natural in its repeat unit, maybe not well defined in its degradation. And also, again, they're kind of exotic structures that maybe are a little bit expensive to think about preparing. Same here with the methacrylate example again. So we took this as inspiration to design something that was maybe addressing these challenges. And that brings me to the polymers and the polypeptides that we actually designed and tested. And so my idea for this came from some inspiration from my graduate work. Actually, I did a lot of work in my PhD on glycopolymers, but also on methionine, which is really my favorite amino acid to work with. I think it's the coolest amino acid because of the chemistry that you can do on it, which I'm showing here. You can take polymethionine or a methionine-rich peptide or protein. You can treat it with hydrogen peroxide, simple, cheap, anyone can get it, and turn this into polymethionine sulfoxide. Or you can treat methionine residues with alkyl halides and turn them into sulfonium salts. And because your R group can be quite variable, you can add a lot of different groups to methionine very, very easily with this chemistry. And some advantages and reasons why I really think that methionine is cool beyond this chemistry is that it's really cheap and easy to think about commercializing. So methionine is naturally occurring for one. So all the building blocks in this polymer would be natural. And, you know, when thinking about degrading it, it's going to turn back into something that the body knows what to do with. But also it's really inexpensive. And so even on the scope of like buying amino acids in the bottle, methionine is quite cheap because it is used as a feedstock in farm animals and is ready to get, or easy to get from agricultural sources. And so, but it is important to note that if we were to take just a polymer of methionine, it's actually a quite hydrophobic amino acid and it's not water soluble. And so if you want to use it in water applications, you need to think about using one of these two types of reactions to convert it to hydrophilic structures. But these reagents are very cheap and easy. And if we were to choose an R group that has a carboxylic acid, like shown here, we will end up creating a zwitterion. And so compared to the zwitterionic structures I showed a couple of minutes ago, this is really attractive because all the building blocks are natural, they're cheap, they're easy to get, And these reagents are also really cheap and easy to get. You can buy like kilograms of bromoacetic acid for not much money. And I wanted to kind of just, in the spirit of webinar and kind of education in the peptide community, wanted to give a shout out to some of my graduate work, actually, where I was working on methionine, because I think these reactions are really, really useful and maybe underutilized and underappreciated still. This is a reaction I did a lot of work on as a graduate student to kind of define the scope and tolerances of this. And it's actually pretty click-like, not completely, I wouldn't say we can call this exactly a click reaction, but it's click-like, these alkylations, because they are quantitative. You essentially convert every residue into the structure that you want. Water compatible, also compatible with organic solvents, but also pH independent, and we don't have byproducts as well. So those are many characteristics of click reactions. And the reason that it really doesn't matter what the pH is, methionine doesn't care. There's no proton to gain or lose. The sulfur is in this thioether is actually just a great nucleophile, and it doesn't have to gain or lose a proton, so it doesn't care about pH. And water is not involved in the reaction either. There's no proton to gain or lose by water. And so you can do this in water, alcohols, DCM, what have you. And you can pick a lot of different R groups, very broad in substrate scope. So in my work in the Deming lab, actually, we showed lots of different examples. Of course, if you have triflates in there, you don't want to do this in water, they'll just hydrolyze. So these in particular, we did in organic solvent, but all of these shown here, we did in water. And so I just wanted to kind of make the community aware of this really easy reaction if you want to, add a group to a peptide or protein that contains methionine. And in some cases, you can actually use this as kind of a click and release strategy, where we looked at the stability of these groups, these sulfonium groups. And so in some cases, they can be labile with this pyridine derivative. And I'm showing in this table some examples of groups that are stable and some that you can remove later with those specific conditions. And there are some applications where that may be useful. But in the case of the zwitterionic structure that we decided to use, it's quite stable. It can resist boiling. You can take that sulfonium salt and boil it. The group's not going to come off. It'll sit there on the bench top. It'll sit there at pH from 2 to 10. Very, very stable. So that's really attractive in terms of using this in commercial formulations. You don't want it to degrade if it gets hot on a truck. Okay. And if you thought that these looked a little bit weird, maybe, but they're actually found in nature. So alkylated methionine sulfonium salts are natural and actually marketed as dietary supplements. And so methylated methionine has been called a vitamin U. It's sold like in healthcare stores, and it's actually found in tomatoes and green tea and some other natural sources, even in cabbage. And I need to maybe email Green Life and tell them their chemical structure is a little funky here, but this is one of the sources. You can buy vitamin U. And also methionine salts are really, really important in biological methylating reactions. And so this is another example of an alkylated methionine that's sold as a dietary supplement. And this group where adenosine is connected to methionine is really important actually in transferring methyl groups to nucleic acids, for example, in epigenetics. But also to methylate proteins, lipids, and other metabolites. And so that's the structure here, and you can actually buy that and eat it. So alkylated methionine sulfoniums are maybe not as strange as they might look in that table I showed. Methionine sulfoxide, that other hydrophilic derivative of met that I mentioned, is also natural and actually essential for life. It's used as a scavenger, actually, for oxidative protection, or protection against oxidative damage in cells. So this is the reaction. We have reactive oxygen species generated by metabolism and other events. And MSR enzymes, or methionine sulfoxide reductase enzymes, will convert methionine that has scavenged these reactive oxygen species back to MET. So totally natural in the body, and actually really important. And you can buy MSR enzymes actually in gray hair shampoos, because part of the aging process is this increase in reactive oxygen species and inability to deal with them. And so we see differences in brown hair versus gray hair in terms of the level of oxidized methionine and the enzyme levels. And I also wanted to point out one of the reasons I was interested in potentially using polymet sulfoxide as a PEG alternative is that there is, granted, old studies showing the tolerance of chains of polymet sulfoxide in rodents as being very, very well tolerated, actually more than 2 grams per kilogram IV, which is remarkable. So that was another indication that these types of structures would be really attractive for therapeutics. Okay, so how do we make them? And in the Peptide Society, I'm sure most of you have used solid phase peptide synthesis at some point, or are very familiar. And then the other way of that's really common, of course, is like recombinant synthesis. My lab uses an alternative technique that has been around for over 100 years at this point, actually, which is, and maybe longer. I should say that the NCA reactions to form peptides may be involved in the origins of life. They've turned up in like primordial soup studies, like reenactments of origins of life conditions. And what happens is an amino acid is converted into a 5-membered ring structure. And the way we typically do this is with halogenating reagents like trifosgene, phosgene, but there's many phosgene alternatives too. So you need some kind of halogenating reagent, and you make this ring structure. And the kind of 100-year-old way, and way that people still use and have done some really nice optimizations on, is with nucleophiles, typically amines. In the Deming lab, we had used zero-valent metals such as cobalt and also nickel complexes to initiate this. They all work really well. The metal complexes do some really beautiful chemistry and have fast kinetics, where the reaction is done in less than an hour usually. So you can grow chains that are even 900 amino acids, for example, in about an hour, which, if you've done solid phase synthesis, that sounds pretty dreamy. And of course, they're not sequence specific. That's kind of the downside, or it's just a caveat. So in some applications, that doesn't matter. And and particularly in the one I'm talking about today. I wanted to point out that the NCA chemistry is used commercially. And so if you've bought polylysine, for example, to coat your tissue culture slides, that was most likely made by NCA chemistry. And then I'll refer back to that table of peptide drugs, top selling peptide drugs on the market today, or as of 2019. The glatiramer acetate example is an MS drug, and it is actually made by NCA chemistry. And I forget, it's a mixture of, I think, aspartic acid, glutamic acid, and a couple others, but it's a mixture of 4 amino acids. It is not sequence specific, but it actually works really well. And it's much cheaper to make and scale than using solid phase. So we know it's commercializable, which is another reason that we like this route, particularly for the application I mentioned today. Okay, so the preparation of polymet CV7 was commenced by making the NCA, making the polymet, and then we did react the amine terminal with this alkyne group. So we have this polymet with the alkyne on it, and then we alkylated or we oxidized using those reactions that I showed you a few minutes ago. It's really easy to do this on multi-gram scale. We have a nice purification method. Any of you doing polymer synthesis know this is really important. And so these are pretty tricky as they hydrolyze, and so we have an anhydrous chromatography method that works really well. And the reason we wanted this alkyne there is so we could attach the CB7. And the reason that we picked this methylated version, which is, for one, it's natural, that I mentioned, like in tomatoes and things, so we know it should be well tolerated, but also we wanted to compare this zwitterionic structure to this overall neutral sulfoxide to a cation. So we attached those structures to CB7. We created the complex shown here, which was incubated with insulin, recombinant human insulin, and allowing this host guest complex to form. Some of the stoichiometries are listed here. And so the experiment that we usually do to test for insulin aggregation is to shake it like it would be on a truck at 37 C. So it's basically like it's on a hot truck, and collect absorbance measurements over time. And right away with mixing this panel with insulin, we saw that, interestingly, that the methylated and oxidized versions of these polymers started forming aggregates right away. So they were obviously interacting with insulin. And that wasn't what we wanted. We didn't see that for the zwitterion. And so this was pretty surprising. And so we identified where we sort of dropped out a couple of candidates right away. And we don't think that that effect is conformational. They all have kind of a similar CD spectra where they look like they're extended disordered structures. This is just a reference of canonical spectra. And so we think, you know, it kind of matches these extended spectra. It might be that the effect is not conformational, but maybe interacting with hydrogen bonds and either to insulin or to water, because DMSO, which is kind of like met sulfoxide, forms really strong hydrogen bonds to water, actually stronger than to water-water hydrogen bonds, and you get linear ordering. So that is possibly an explanation, and whereas zwitterionic structures don't seem to do that same thing. So that's why we think those two candidates were not the best for interacting with insulin. However, we were really happy to see that, like some of these prior zwitterionic structures, our zwitterionic CB7 insulin conjugate was really effective. And we tested a whole bunch of different chain lengths from 12 to over 300 units. And we found that 12 residues is too short. 25 is actually stabilizes over time pretty well, but by 80 hours of shaking, hot shaking, you start to see some aggregation. So somewhere between 25 and 80 is the sweet spot, because by 80 residues, we could keep shaking that insulin for 100 hours and there's no aggregation. So this was really beautiful data that we were really happy to see. We wanted to verify that this wasn't just unique to insulin. So we looked at another protein or a peptide that will also aggregate. This is calcitonin. It doesn't naturally have an aromatic at its N terminal, so we put one there. And this is the structure shown here. We used two different linker options to see how well that would fit inside that CB7 cavity. And so this is the sequence of the human calcitonin here that I should say at the top. And so we looked at the aggregation effects again. And what we saw was that, again, all alone, calcitonin will aggregate like crazy. And that if we don't have the CB7 there, it also doesn't work very well if we just put the therapeutic and the chain in there, but they're not in close proximity. That doesn't work very well. But the conjugation to the CB7 and really bringing that therapeutic and that zwitterionic chain close together really does inhibit aggregation again. And we found one of those linkers actually allowed, better conjugation. And so this one in blue is the A2, and that is the 2-carbon chain here. So just a minor detail that allows it to form this complex better. But what we can say from this data is that the zwitterionic polypeptide works well for insulin, calcitonin, and probably a lot of other peptide therapeutics that aggregate. And now a little bit of data just on what that zwitterionic structure might provide us in vivo. We haven't tested in any animals so far, just in cells, so that's future work. But we can say in cells that structure, as expected, is not toxic at all, even up to 5 grams per liter dosing. And over time, it will degrade by proteases. Actually, papain was the best one. We started to see slow degradation by 1 week. So because of that zwitterionic structure, it's not a common amino acid, right? We do see these alkylated mets in nature, but not in the abundance or sort of density that we have on this chain. So the degradation was slow, which I think is good in some cases. If you want the sequence to degrade faster, we could simply add a few other amino acids as cleavage sites in the middle of that chain. Like if we add some alanine residues, I guarantee it will be a much better substrate for some of these proteases. So we could tune the degradation, which I think is nice. Okay, so a summary of this zwitterionic work is that these peptides are a really promising alternative for PEG as stabilizers, and I think likely for self-coatings. We do need them to be longer than 25 residues to have that effect, but somewhere between 25 and 80, we'll have to hone that in. I'm guessing, maybe a 30, 40, or 50 mer will be the sweet spot. But the carboxymethyl sulfonium that I showed it's really cheap and easy to prepare. Even, a brand new undergrad could do these reactions. It's not cytotoxic. It will degrade with natural proteases. And a key feature in this is using the supramolecular host-guest chemistry with CB7, where we can simply have an N-terminal aromatic group, bring the zwitterion and the therapeutic in close proximity, and we'll get this effect. So if you want to keep updated on these projects, you can follow me on Twitter, and I usually post when we have a new paper. So I know we're almost out of time. In the last couple of minutes, I just want to briefly mention a related project, which is really cool, and give a shout out to some other stabilizing polymers that we're working on in the glyco field. We need thermal stabilizers actually to withstand freezing conditions, kind of the opposite where I was talking about heating. Formation of ice crystals is actually really damaging to biomedical tissue samples, cell tissues, organs, but also even in like ice creams. No one wants crunchy ice cream, and also cracks in concrete. So ice crystal formation, we need stabilizers actually against the other way too. And my lab has been thinking about structures for this. We have been inspired again by nature, just like those alkylated methionines. We've been inspired by glycoproteins made by Arctic and Antarctic fish, and also some mountain ticks. These are really simple repeats, and they can be anywhere from very short peptides with 4 repeat units to longer, kind of bigger macromolecular structures with up to 50 repeat units. And they're really simple: alanine, alanine, threonine repeats with glycans. And they have this kind of extended PP2 structure. And these are really of great interest because they protect extremophile organisms from ice crystal damage. So there's been a lot of interest for commercial applications, but as you can imagine, polar fishing trips are not practical, and extracting these from fish blood is entirely impractical. So we've been inspired to kind of think about these ice binding properties and harness them with our own materials. This is how these AFGPs, or antifreeze proteins, work. They bind to the surface of ice crystals. You can actually see fluorescently labeled ones here. And over time, the ice crystal growth by the surface binding is inhibited, and you get much, much smaller crystals than you would in untreated samples. And this is an example actually from ice cream. And again, the effect is length dependent, just like I showed with the methionine structures. The longer chain, more repeat unit structures are more potent in this activity. So this is some recent work that a fantastic graduate student in my lab was the key person on, Dr. Anna Delaray. She is going to be on the job market soon. If any of you are looking for a talented materials, polymer peptide glycochemist, she has many skills, so keep Anna in mind. Her manuscript is under review right now on this work. We use the NCA method again to synthesize synthetic anti-freeze glycoproteins, scalable, rapid, much more accessible than a polar fishing trip. Again, they adopt these kind of native conformations that we see by circular dichroism. And the really cool thing is that they shape ice crystals just like the native fish proteins. There's a few examples here showing length dependence, and also it depends what sugar is attached. What it's really nice is that the native sugar is more potent, but we can actually use much simpler sugars and still get strong effects, which is really cool and something people have kind of argued about. And this is an example of how we see the ice crystals forming over time. You can see in this native threonine to alanine ratio, crystals are really, really small over time. And so we don't get ice crystal growth. This is perfect for biological samples or ice cream. And again, the longer chains are more potent, just like we see in the native fish proteins. And we can use simpler sugars, and it's really quite potent. So again, keep an eye out on Twitter if you want to learn more about this project when we get through the review process. But we're pretty excited because we've made some nice discoveries about what parts of the structure are important, and that they have very native like activity. So those are two examples of peptides that are stabilizing for many applications. I'd really like to thank my entire research team and our funders. And I would love to interact with you all for whatever questions that you have today. I don't see the Q&A now, so I'm going to rely on Kevin and Lauren to let me know how to see them or to share those questions with me.
Yeah, let me take a look here, Jessica.
Okay.
And I may need Lauren's help. So I'm not seeing anything in the Q&A.
No, I don't think it's right now.
Okay.
Don't be shy, you all. There's no dumb questions. If there's students watching, please speak up.
Yeah, that was and really exciting stuff. I have so many ideas for some of the projects that I'm working on that this might be helpful for. So I do have some questions. One, so what is the longest? Do you have thoughts of looking at much longer peptides or larger proteins? Obviously, you mentioned mRNA vaccines, so it must be on your mind.
Yeah. So for the zwitterionic polymers, yes. For both of these structures I showed that are stabilizer molecules, we're really interested in kind of thinking about, yes, both antibody and antibody drug conjugates and mRNA vaccines and things that are stored cold. It's really expensive to ship these things, and when those conditions fail and we lose these materials, it's a huge loss. So that is definitely part of the future directions, is to expand this beyond insulin.
Yeah, great.
I actually have a question. Can I ask that?
Oh, yeah, go ahead.
So you talked about the protease, we're testing proteases. Are you thinking of using these for drug delivery at all? Like, do they cross over cell membranes? Or is it...
Yeah, that's a good question. So yeah, in terms of like drug delivery, for example, with insulin, you would inject the complex and it would stabilize the insulin while it's in the vial, shipping and shaking and all that. But once it's inside the body, they should dissociate, and there's lots of good data on CV7 PEG conjugates. So we know that should happen. And then what happens to the polymer then? And that's exactly why we don't want to use PEG. And so we, with the structure we showed there with the CB7 conjugate, we have not tested intracellular localization with that one. With some other alkylated, actually glycopolymers where they had the sulfonium group and a sugar, they go into cells like crazy. And so that is really interesting, and we don't know exactly how that happens. We did some work with Gerard Wong's lab at UCLA, and they looked at SACs, their X-ray scattering, and looking at how the membrane of the cell bends and interacts with the structure. And it looks like it actually, without using receptors or any kind of endocytosis, it actually just causes a buckling in the membrane, and they would pass through. So it really does depend on exactly what structure you use. I can also say with these antifreeze proteins from the fish, with these sugar structures, they also get into cells really well. But it is endocytosis driven there, and we have to do a lot of work to figure out what kind of receptor that's at play, because we know when we do those experiments at room temperature or cold where endocytosis is inhibited, you don't see entry anymore. So I think, like, you know, any kind of new polymer, it's going to take a fair bit of work to understand the details of the biology. But we're really happy that even when they go into cells, they're not toxic to high concentrations.
So you want them to get degraded. Like that would be eventually the goal, is that you want them to break down. Not,
yes.
Because I know a lot of people are trying to use like D-amino acids or something other that would be less likely to be degraded. This is kind of the opposite.
Yes. You're yes, absolutely. And actually just maybe a week ago, another postdoc and another student in my lab, we published a whole paper on using D-mucin. They're like D-mucin analogs, where in that case, we really don't want them to be degraded, because part of the therapeutic value of artificial mucus as lubricants and things is that you want it to last a long time. Like, you don't want to put eye drops in your eye every hour. You want to put them in there once. So in that case, we don't want degradation. In this case, if you're thinking about something like dosing frequently for insulin or maybe some antibody drug conjugates, you want those stabilizers to degrade over time because you're dosing very frequently. So it really depends on the application, but luckily we can tune those properties. Yeah, great questions.
So here's a question. So speaking about like circulation time, do you think that the increase in circulation time that you mentioned is long enough to make this effective as kind of a long-acting release delivery? And how long would that actually be?
Yeah, that's a good question. So with the zwitterionic structures that have been compared head-to-head with PEG, the circulation times were quite similar. We haven't done, I mean, this is pretty recent work where we just did in vivo, or sorry, in cellulo kind of, or in vitro assays. And so we don't know with this particular structure. And that's again something, you know, it's a new polymer. We're going to have to kind of do these head-to-head comparisons against other zwitterionic structures. But if they perform like some of these kind of better studied structures, they'll be really similar to PEG, probably a couple of, you know, a day, on the order of a day or two before they're fully excreted. But again, you know, it may be dependent on these exact structures, and because they're based on peptides rather than, you know, kind of plasticky polymers, it could be different.
Okay, interesting. Yeah.
Yeah.
And here's another question on the alkylated methionine. What is your opinion, based on your background, on how effective that is for health? Vitamin
the vitamin U.
Yeah.
Yeah, I guess with the methylated versions, I'm not sure. I really don't know enough about that biology, but I can say with the ones with the SAM or SAM-E, there where there's the adenylyl methionine, they seem to be really important for these methyl transfer reactions that are essential for epigenetic changes and those kind of signaling events. And we know that that's going to be really important for when genes turn on and off. And if we don't have the reagents in our body to like conduct those reactions, I think we're in trouble. So probably good, but you know, I think the old chemist saying is the dose makes the poison. So I don't know if supplementing and getting really high levels in your body more than what we would get from like a healthy diet that includes tomatoes and cabbage and all those things. I don't know. So my opinion on that is probably just to eat your vegetables. Yeah.
We'll keep that in mind.
Yeah.
So those are all the questions I see coming in. If anybody has any, you can obviously type them in the Q&A really quick or raise your hand and do it live. Otherwise, we will conclude today. I think, Lauren, you have a final message before we do that.
Oh, I just wanted to one last invite for everyone to come to the meeting, and that includes you, Dr. Kramer, if you want to come.
Thank you. I would love to.
September 18th to 21st, so it'll be fun.
It's in Napa Valley this year.
Yeah, I just think it'll be really nice. We have a couple social events also in addition to the conference, so it's a really nice networking meeting, I feel like. So
well, yeah. Hard to turn down a trip to Napa, so I'm sure you'll get a good crowd.
Thank you for coming. Thank you for putting this together, Kevin. And I think that's it.
Great. Thanks, everyone.
Well, thank you again for the invitation, and look forward to interacting more in the future.
All right.
Definitely.
Okay.
Bye.
Dr. Kramer.